All right. Hello, everyone. Welcome back. Thanks again to our incredible speakers for these talks. I was learning so much today. I was writing notes to myself all throughout. And thanks to those who could make it. I know June Round wasn't able to make it. She had other things to do. But we're excited to have all the rest with us to chat a little bit about this field, about the field of microbial metabolites and human health, which I think has been growing very, very rapidly in the last few years. I'll tip it off to you, Sid.
Yeah, well, I just thought I'd start off. Well, first of all, thanks for accepting our invitation, and what a wonderful set of talks. Yeah, it was amazing to watch. So I thought I'd just start off with an icebreaker so that, we get our conversational juices flowing, so to speak. I was wondering, and this might be something which is also would be useful for grad students out there or postdocs. What motivates the six of you? I mean, we personally admire you and your science very much, but what's the driving force in your lives, especially at a time when, things are admittedly a bit uncertain? And anyone can join in. This is not a quiz, so feel free to interrupt and have conversations.
Yeah, I'll just jump in. I would say stubbornness, particularly at this time. So I mentioned I'm nearly 60. So, one of the things I tell all the junior people that I talk to is, no matter what's happening, and this is a particularly difficult time to do science, but no matter what's happening, you can't give up. So there's just a level of stubbornness, but also just, the questions are so intriguing and they're so sort of open-ended, they feel expansive. So I think that's a thing where, we get excited to dip into like a whole new arena like endobiotics. So I think it was Crystal who said, there's always more questions in science, and that really is the motivator.
Maybe we'll call on people. How about the next Matthew?
Um, yeah, I think I feel very similar to the sort of latter sentiment, maybe especially for me. I'm still quite early in my career and I'm still kind of like uh, you know, uh leveling up, so to speak. And I just still feel like I'm learning so much and starting to get a better grasp on like uh, you know, especially with forums like this, like you kind of start to get a sense of the questions that everyone's asking and the things that like people need, both as like scientists trying to solve questions, but also like, you know, you know, in terms of like health challenges faced by people that like our field is very poised to address.
Thanks, Matt.
Yeah, maybe Brittany. I'm going by order on the screen. Yeah, the prior answers make me think a little bit too of like basic versus translational stuff and how the funding climate is changing and how our motivations might be shifting as well in this climate. But anyway, Brittany, I'll throw it to you next.
Yeah, I would guess that all of us have, like, at the heart of our motivation, though. I mean, we all need funding and we all need whatever the political or financial realities of our jobs. But I don't know. The thing that really motivates me is our job is fun. It's like interesting to be able to go to work and think of what do you want to find out and how could we try to find it out. And I don't know. A lot of people don't have that luxury and privilege in their career or their job or their day-to-day life that the thing I think is fun is also my job.
Great answer.
Privilege.
Yada, maybe you can go next.
Yeah, I would say all of the above. As I agree with all the previous speaker, it's the curiosity of I need to know what's happening, and which goes with the stubbornness, right? And the need to learn. I don't think there are so many things I don't know that I need to know, and it's fun. So all of the above.
Last but not least, Matt Melody.
Well, I always think that I'm very lucky because my job is something that I really enjoy doing. So it doesn't really feel like a job. So I'm very fortunate in that regard. And I always wanted to do something that would be meaningful for my life. And I probably just happen to be good at studying the immune system or being a scientist. And as far as working on gut microbiome, one biggest motivator for me was I had GI issues when I was a graduate student, and then I got into microbiome um, research partly because of that. And then now I realize the gut microbiome is actually a really fascinating area to study because it will offer a lot of potential therapeutic targets that would be easier for therapeutically to um, either supplement or fine tune or modify by diet. So I think this is really a very exciting field. I'm very fortunate that I'm working on microbiome.
Well, we are very fortunate that you are working in this field. One of the nice things about this conversation is that I get to put this guy in the hot seat. What's your answer to this question?
Oh,
What motivates you?
well, I think I agree with everyone, and essentially, I think it is fun and joy, and it comes across in all of your talks that you actually really are passionate and interested in what you're doing, and that is you can't take that for granted because most people and most jobs don't have that. So no matter how dark the time is, having our trainees and students see that joy, I think just sort of justifies the difficulties that we might be going through now. How about yourself?
I mean, this is a very uplifting set of thoughts that all of you. So even if I was even remotely depressed today, that would have been dispelled. Um, I just, I just want to do science for the benefit of humanity, and that's how I feel like I learned that at Rockefeller where I did my PhD, and I've just never changed. And of course, things are very difficult and uncertain. So really appreciate everyone's thoughts about this. And I think it would actually be very useful for the youngsters out there, or even
Yeah,
old farts like us. Yeah.
but to that point, I guess maybe going on to the like a science
Yes.
sort of in question, and I'm actually changing a little bit from what I said I was going to talk about, but
Uh-huh.
this makes me think about clinical translation and some of the critique that we get in the microbiome field. I hear a lot of people from other fields, especially saying it's a big waste of money. We haven't translated much out of the microbiome research. You have fecal transplants, and what else is there, right? It's like what's the promise? Um, I saw in almost all of your talks at least some kind of preclinical validation, or even starting with human intervention trials, or moving into clinical validation trials in this space where we're leveraging the kind of functional outputs of the microbiome rather than just paying attention to the ecology of who's there. I think that's really where we're seeing a lot of the rubber meet the road in terms of finding practical ways we can manipulate disease states in the host that will lead to, approved therapies down the road. But, how do you all feel about, have we not lived up to our promise? Are we going too slow and wasting everyone's taxpayer money by studying this area? Or is there huge promise and therapeutic potential? Any hot takes?
Go ahead, please.
Jetta.
Uh, I don't know. For our case, for example, I think my feeling is that we are too slow. However, we are moving. I wish we were moving much faster and we could, provide a solution for patients much quicker. But I feel like we are slowly progressing from the preclinical data to actual clinical practice. Um, so if we had, probably if we had much more funding, we would be much quicker. But funding is limited as well in Canada. So maybe we are in less darker times than you, but still funding is a limitation as well for translational science.
Yeah, that's good for all the funders who might be watching today. Give us more money.
That's right. It's also not easy to run clinical trials without an MD.
Mhm.
I mean, but of course, we can collaborate always with others. Anyone else have any thoughts about the issue that Sean had posed?
Yeah, I mean, I feel like it's a fair question for the folks that are paying the bills to ask. But it's a little bit unfair because, I'm old enough to remember that when we were going to sequence the human genome, that was going to solve all of our problems. We were going to have all these targeted therapies. And it certainly has been a huge help to have the genome sequenced, and to continue to sequence it and continue to look at it. But the level of complexity that we're dealing with is so much bigger than that. So much bigger than that. So in some ways, it's a little unfair to put our feet to the flames and say we ought to be putting stuff over the line, therapeutically. And I guess I think about it from the point of view of, say, a big drug company. If you call a big drug company and say, hey, why don't we target some of the stuff that the human doesn't encode and is different in everybody? They're probably going to hang up on you. And I haven't had that exact conversation, but we've tried to translate some of this stuff pretty far. And I think the decisions at the end of the day, while disappointing to me, were the right decisions because we're trying to solve problems for drugs that are off patent. So where is the motivation in the US system to do that? Interestingly, we've gotten some interest from the EU where it's managed care, and some of the drugs we focus on are used widely there. And I would be thrilled if something translated into the EU practice where the profit margin isn't so such a driver. So I sort of understand the difficulty of getting things there, but I challenge the assumption that it should be easy. Because, we're just dealing, it's not even GPCRs is hard. And people did that for decades before we got a lot of drugs that worked for them, but we didn't design them to work for them. We figured out they worked because they hit GPCRs, So starting from a view of sort of, we don't really know what the microbiome is doing to, we figured it out, we can drug it and control it in every human in a big clinical trial. That's a lot of things to line up.
Yeah, I like to use the example of like prior fields where basic science is translated to the clinic. And if you look at immunotherapy, it was like 50 years between the initial discoveries and the therapeutics. And for mRNA, which is like a very fast turnaround, right? It was like 15 years between the discovery of the uracil modifications and vaccines going into arms. That's about the amount of time it took to get to FDA-approved fecal transplant alternatives, right? So I think we're actually moving about the rate most fields move into therapies.
Yes, Melody.
I totally agree with what you guys said. And I also want to highlight that the human microbiome project started in 2007 or 8. So it's only, less than 2 decades. We've made tremendous progress. And now if you talk to everyone knows what microbiome is. And we are bringing more awareness to, how important diet is. And I think we are actually making tremendous progress. And I agree with Matthew's point that the microbiome is really complex. The complexity would require tremendous technology for us to really map out what's coming from this bug and then what immune cells are being affected. And now we've gotten a lot of tools established, single cell transcriptomics, metabolomics, lipidomics. I think now we're in prime time to make really tremendous discoveries in the next decades or so. And I think it would be a shame that we're losing the momentum.
Any last takers on that question before we move on?
Yeah, I mean, I think it's like people have kind of touched on it. Obviously, there's like maybe you could be construed as like waste in the public sector, but also there's just been a few $billions poured into different biotechs at this point, and there's like 2 FDA-approved therapies to show for it. And I think it really is just like there's still like a form factor that people haven't figured out. Like there's been a lot of different like modalities that people have tried. Lots of like microbiome therapeutics companies kind of try to fall into the same traps of like maybe it'll just work this time. And I think that's just like not very wise. And so I think it's just a matter of because of the complexity, because of the microbiome, but also, the diseases that are involved that are associated with the microbiome are themselves very complex. I think it's just like um, that's kind of the task, is to figure out what the drugs actually look like. Like right now, the best we have is basically FMT, which is kind of like, it works in many cases, but it's still us kind of shrugging. Like maybe it is to some extent. A lot of these, maybe the drugs are actually more like kind of like other Matthew's work, where it's like you're understanding the like microbiology involved, but actually maybe the drug doesn't necessarily have to be a microbe. Maybe it can be like just understanding the microbial pathways involved, and how it interfaces with the host. Yeah, to me that's the challenge.
Yeah, and like maybe one last point I'll bring up is, what do we call a drug or an intervention, right? If you're talking about FDA regulated stuff, that's one side of the equation. But in the microbiome space, if diet is manipulating the system and the outputs of the system, that's never something you can drugify, really, right? So we know a lot about dietary patterns that are mediated by the microbiota that have health effects, and I think there's obviously the Sonnenberg lab has done a lot in that space. So I think we should take those as wins as well. Anyway, Sid.
But just to sort of, bring this to a close, I just want a quick answer from all of you, including Sean. Namely, um, the question which I have is the mechanisms that we are, defining using the reductionist experiments that we are doing, that pretty much most of us are doing at least. Is it just that they are not deep enough? Is it just that they are not translatable across humans because of the variability? We've all touched on concepts such as gut barrier permeability, education of the immune system, signaling to the brain via, enteric neurons or, systemic circulation. So we have all these general principles which I feel have come out of studying gut biology. But then the point that Matt had raised, that we only actually have two drugs on the market. The question is, are we just not doing enough on the mechanism side, or is it time to make a hard pivot and do more human studies to try and understand that variability? And maybe just a quick, 30-second, enough or not enough.
So I'll take this first. I think when you talk about the gut microbiome, I think you're dealing with an incredibly plastic system, much more plastic than typical things that we try to drug in a person. And so, diet's a good example. Here's something we do multiple times a day. We put sometimes very different things in. The system adjusts. So it's pretty incredible that way. And so I think, you think about the things that like recurrent C. diff, terrible situation, FMT is working, that's great. That was probably the low-hanging fruit. And thank God we have something. I think if you look at IBD, so complex. I mean, IBD is going to be like breast cancer, where it's not one disease, it's not two diseases, it's dozens, if not maybe more than dozens. And so I think it's a little bit hard to use the same lens that we look at, a family of nuclear receptors, a family of GPCRs, even chromatin remodeling factors. Those are still more defined and less plastic than I think what we're dealing with.
Wonderful answer.
That's a great point.
Yeah.
I think also just as mechanistic preclinical studies and in vitro, any lab studies that happen, as that continues, like there's momentum growing for mechanistic and causative studies, that's going to enable us to do a lot more human studies than before. Because, 10 years ago, if we were all supposed to come up with a clinical trial, it'd be like, well, an FMT, I guess. But now we have a lot of mechanistic things we can design targeted human studies where that cost and kind of the input of all the labor to do that kind of study is worth it when we have more mechanistic things. You asked if are we doing enough? I think it's just going to naturally go that direction. The space is big. There's a lot of people. There's plenty of people to cover the different elements of it, I think.
Yeah, that's the great. It justifies the human trials essentially to have
Yeah.
that. But Matthew, oh, as Melody, go ahead.
Yeah, I just want to add that I think a lot of the studies that have been done, probably mostly just by giving the metabolites or probiotics and hope for the best, hope for the results that you want to see. But at least with some preclinical trial studies in my lab and some other people's lab, I think it's not as simple as giving the metabolites and hopefully the metabolites will reach the cells in the lungs, in the brain, and induce the changes that you want to see. What we've learned is that for diet studies, when you give a certain diet to the mice, let's say high fiber diet or patent, what we see, at least in our lab, is that you will see tremendous short fatty acids in the gut lumen, but we could not detect it even in the lung, bronchial alveolar lavage, or even in the plasma. So it means that we're not really delivering the metabolites to the organs that we intend for the metabolites to induce the result. I think that would be something that needs to be resolved for human trials so that we can see the positive outcomes that we want to see.
Thank you.
Yeah, totally. I mean, that's the biggest problem any pharma company tells you about if they already have a drug candidate. It's like, how do you deliver it to the right place for the right duration and all? Yeah, that's very, very difficult.
Yeah, and I really appreciated Matt's point about, the fact that we need specificity. Broad acting therapeutics are of no use. So, yeah.
Yeah, and maybe I'll jump in with my take a little bit here, which is that I
Please.
love these deep mechanistic studies are always very beautiful and elegant, and you get a clear answer in like a mouse system or a C. elegans or what have you. But I usually work with really messy human population data, where the causality is very unclear, but at least in that you have the full dirty context of how that metabolite's behaving in the wild. And so, Matthew Carter was talking about, Sonnenberg lab and that sort of vision of starting with the human population, seeing if the phenotype's robust, and then backing towards the mechanism. For a lot of mechanisms, there's probably 10 different mechanisms that give rise to the phenotype. In the paper that's published in Cell shows you one, but there's probably a few other ways it happens in the context dependencies. And so, I think going from the complex real world and then back to mechanism is a really fruitful avenue to understand heterogeneity and like which causal path is most relevant to treating a disease.
Except that to publish a paper which goes back and forth between humans and animals requires an incredible amount of funding. And that may or may not be something that's feasible for young investigators, which is a wonderful segue into my next question, which is that, the space that we're all occupying is, it basically, there's mucosal immunology, there's gut microbiology, there's, like Brittany, a hell of a lot of neuroscience. And so you have a convergence of all of these fields. It's not easy for young people to know how to get into something like this, without tons of funding and so on. Should people like that start off by just analyzing publicly available data sets and get into, how does one actually start working in this field? Can, a young investigator, do they need to have a fully functional germ-free facility and a high-resolution mass spectrograph and a Nextera sequencing, or is this area really something that can only be feasible for big labs with tremendous amounts of funding?
Well, we've never been big and ever had tremendous amounts of funding, but we take, unlike Matthew Carter, which I love the top down. I mean, it's just it's so important to take that approach because then the mechanistic studies get really focused and are immediately relevant. but we've spent a lot of time looking at extant data. And even, a relatively small lab like ours, when we publish a metagenomic or proteomic, we only analyze just a small fraction of all the stuff we put up in the data set that we report. And so if you take the time and you look for it, there's gonna be information in the stuff that everybody else has put up. So I wouldn't have thought of it, but Sid, you said, yeah, start with what's out there. Because most of the time, and I think, the cell paper, as Sean said, the cell paper may only have one mechanism. There's probably several others. It's going to be true of the big omics data as well. So I would think that's a way to get into it before you really spend a cent in the lab.
Yeah, and this is especially useful for, even young PIs thinking of wading into new territory where it's going to be difficult to have part of your startup being promised towards these new experiments. Might be easier to just do computational analyses. I don't know. Anyone else want to leap into this? I'm being very selfish here by basically asking for advice.
I can probably add something. I totally agree that there's so many really remarkable studies out there, and there's tremendous data out there that can be leveraged into your research program, and you just need to look for it, and also sometimes collaborate with the right people. You don't have to do everything on your own, and then your skill set might be beneficial to other people, and vice versa. So I think now is really critical that you develop really fruitful collaborations, leveraging everyone around you or people that can be easily reachable. I think really nice that now we're really doing the symposium virtually. And usually now, actually, I would just email someone who might have something that I need, email them and see if they respond. And then if they responded, I will have a meeting with them. And then I try to see if they would be willing to help. And if I could help with anything that they do, I would do that also. So I think I'm very fortunate that I started my lab during the pandemic in some way, because that's a time when we will start doing a lot of virtual meetings. So it would be easier for young people to develop collaborations this way. And I also agree that microbiome work can be very, very costly, especially like my lab, we do a lot of mouse work. But now I think there's a push for using organoid systems. I think that's something that can be considered by young investigators. And the organoid systems are really variable and can help you identify really critical metabolites and signaling pathways in your organoid system. So I think that's something that could be taken advantage of, because now there are a lot of organoid systems that have been developed.
That's a great answer. I love that.
Yeah, the pandemic point I think is great because, we all have our core competencies, right? And that's going to define to some extent what we're going to pitch as our first thing we're going to do as an assistant professor. But as a more computational person, I'm an unabashed data parasite. I love using data sets that are already out there. There's so much juice to squeeze still from all of these data sets, and you can really run a pretty low overhead operation just reanalyzing other data. And during the pandemic, I think those who could pivot in that direction were actually quite productive, right? And a lot of the strictly wet lab folks suffered a lot from the pandemic. And now with the rise of these LLMs and vibe coding and giving access to a little more computational skills from people who don't necessarily have a lot of that training, I don't know if the equation changes a little bit there for people, if maybe this type of an approach is more accessible to more people.
Yeah, thank you for that. I think I feel thank you for answering that question. I think you had something that you had
I have a
wanted to-
provocative one to another kind of hot take that I heard recently, actually from Peter Dorestein, a metabolomics guy, and he was given a talk and he said, doing studying microbial metabolites in host systems today is kind of like reading a book where you don't know 90% of the words that are in the book. However, when I asked a question after the talk and kind of poked him and said, well, you look at the frequency distribution of words, And very few number of words are the most frequently used words, and most words are very rarely used. So if I took 90% of the sparsest words from a novel, I probably could still understand the plot of that novel, right? So I guess my question to everyone is, to what extent in this field do we need to focus on the discovery of unknown microbial metabolites that may be having some effect that we don't yet understand? And to what extent can we leverage our knowledge of existing metabolites, microbes, and genes to have a clinical effect in human populations? You know, what fraction of our effort should be placed in these bins?
A couple of comments while you're collecting your thoughts. Number one, we do very much admire Peter Doherty, both of
Yes,
us.
and I agree. He found all these cool like bile
Yeah.
acid conjugated things.
Oh yeah, no, absolutely. And
Yeah.
you know,
Oh, because my hot take was going to be I hate Peter Dorsey. No.
no, this is almost like the kind of chats that we have over a beverage. So you're sort of being included in these running conversations that he and I have. But yeah, please go ahead.
So I'm not going to go first this time because I keep going first.
Maybe Giada, you haven't spoken in a while. You wanna weigh in?
Yes. So I love Peter Durstein as well. And I always fear when a paper is coming out from his lab because suddenly they have found metabolites or new metabolites that do new things. So, so I have to say it's fantastic and we need that. But on the other side, I fear it because then it might have implication that I haven't thought about it for my work. And so, yes, of course, we have to go and discover everything that is not known. But I feel like we know so little of what we know that for me, at least I need to know a little bit more of those two, three molecules that maybe are part of a bigger picture and play a little role.
But if I may just pose this question while we are having this conversation, within the ladder of known classes of metabolites, you know, short chain fatty acids, secondary bile acids, all the aromatic acid catabolites, the indoles, and so on. It's quite hard because it seems like the short chain fatty acids, of which this guy is a world-renowned expert. You know, it seems like they get a lot more importance. Is it just because we've known of them for a long period of time, and so the if bi biological effects for some of the other classes are just less well known? Brittany?
Maybe. If I don't know if this was a rebate to
Oh, please.
maybe. So maybe I feel like yeah, maybe those are better understood because the bacteria are have been investigated more that produce those short chain fatty acids. I don't, I'm gonna leave this to the others.
Yeah, maybe this goes back to the words in the book thing, right? Like
Mhm.
short chain fatty acids are like and or the, right? They're very frequent words in the book. And if you do a first approximation, the gut is a fermenter that makes organic acids. So by mass, they're just a huge amount of what's there. And so they smack you in the face when you look at the system. So you're going to study them probably first. So I suspect we're missing things that are rare, but may still have a high biological effect. But most of the things that are abundant and have a large effect, we've probably covered. But I don't know, maybe others think differently.
Well, I guess for me, when I think about these things, I sort of look. I agree with Giada that I do worry when I see published papers because I'm like, oh my God, is there going to be another 200 compounds that I have to worry about now? And learn all their pathways. And certainly that's sometimes what you have to do. But when I think about something like a bile acid or a secondary bile acid, it sort of makes sense. Like first law of mass action, they are present in high concentrations. Same with short chain fatty acids. There are known host receptors that will respond to this, so you're sort of checking the boxes along the way. And given a field which is maybe a massive ocean, you are looking for buoys along the way to sort of chart your path. If you just dive in and say, I'm going to figure out everything that's in this ocean, wow, I think that could be interesting, but you might be down there for a while. So And I think it's a little bit unfair. We started part of this conversation saying, hey, we should get to the clinic faster. And now the conversation has turned to, by the way, we should figure out every metabolite that's present too, because there probably is a lot of gold in there. And both things can be true, and they're true here. So I guess for me, I tend to gravitate towards the things that I think mass action, known effects, colonocytes love short chain fatty acids. That's got to be important. Known immune modulators, that's got to be important. So with a limited amount of ability to approach things, I have to winnow them down. So I don't disagree with Peter at all, and I have to go on record. Of course, I love Peter Dorstein, but I agree with Sean with your counter, which is if you read Finnegan's Wake, you're going to see a lot of words in there that are only in Finnegan's Wake. Okay, so they're officially words, but they're not necessarily words that have been used sort of anywhere else in the semi-English language.
Yep.
Matt Carter, do you have anything to declare?
I think we're well past the point of diminishing returns. I think for every new amino acid conjugated bile acid explaining some phenotype, there's 100 papers where butyrate's explaining the phenotype. Like, I think it's just kind of the reality. Sometimes it kind of sucks for us when we're doing a study and it's like, oh shit, it's just butyrate again. Like, it's like, I think that's just the reality. I totally agree with the like mass action sentiment. And it's just the reality of like, the more rare these things are, the more low concentration they are, just physiologically, the less likely they will matter in terms of like hitting receptors or what have you. Like, you also kind of have to view it from the lens of like these things aren't bouncing around in a vacuum. Like, humans have been living with microbes forever. Animals have been with microbes forever. And so of course there are like hooks that our body uses to like sense the state of the microbiome. And of course they're going to be the most abundant things. So yeah, I think we're well past the point of diminishing returns.
And what one like wrinkle to throw into the known things, right? Like, I mean, several of you have shown this in your talks. I think Matt Redenbow, I think you had there's not a linear mapping between the abundance of a gene in the DNA and like the sort of a function. And so how do we take microbiome data and map it to the functional outputs? And short chain fatty acids are a good example. It's not just whether you have butyrate production pathways in your microbiota. It matters a lot what you're eating, what your diet is, what are the other taxa that are cross-feeding those butyrate producers. And so all those context dependencies have to be kind of integrated in order to, even for these known things that we want to be able to manipulate and turn up and turn down, it's a complex task to be able to tune that knob. Yeah.
Yeah, and I mean, we deliberately focused our symposium on host effects, but of course, microbes have all sorts of effects on other members of the gut microbial communities. And, that might be a topic for another symposium. But yeah, I mean, that is certainly those cross-feeding interactions are so important.
Well, and they're hard to do. I think Matthew Carter pointed out you can't put Balafala in a germ-free mouse and colonize it, right? And that's something that I wouldn't. Now I get it. We've worked on biosalt hydrolases, and that makes sense. But the technology, you have to work with the technology you have. So germ-free mice, abiotic mice, they've been a huge workhorse, but they're also not normal. They don't have a fully functioning immune system. A little bit of our work has shown they don't have the sort of endobiotic homeostasis that you would typically expect. So some of the platform on which we've built our knowledge base, also, you have to understand where those assumptions have been made. And so I think it gets difficult to sort of advance knowing that we're also built on something that has made its own assumptions along the way.
Great. And maybe if anybody has any final comments on that point, go ahead and jump in. But I'll say quickly, we paid short shrift to the audience questions. We have less than 10 minutes left, but we'll start getting to those now as opposed to him and I just talking. So if there's no rejoinders, I will jump right in. Good. Okay. So it looks like there's a question from Marcia Denise about systems outside of the gut, right? We talked a lot today about gut microbes. What about the vagina, the oral cavity, the skin? And where do you see promise for, having effects on all of these systems of the body by modulating the microbiota of those other compartments?
Also keeping in mind that animal models may be in equally short shrift for some of those, such as the vaginal microbiome.
So I hate to keep jumping in here, but I will. So
We
I've been
love it when you do.
lucky enough to have a basic scientist who then trained as a dental surgeon and as a dentist, and he's been kind of a fellow in my lab and now has his own lab. And so I learned from him that the oral microbiome is actually quite incredible. And in the oral space, a lot of disease appears with biomarkers in an easy place to do sampling, a much easier place to do sampling. And so I actually think, and all the ones you listed are important, of course, but I'll just put my hook in that, right here, most of it's aerobic, but a little bit underneath where the gums are, it goes anaerobic, and all of that is relatively easy to sample. And so there's plenty we can learn there. And you can look at my papers. The person I'm talking about is Adam Litzen, who's here at UNC, and he could give a great description of all the different disease states that present very unique things in the oral cavity, where it's easy to sample, it's easy to get information, and to me that's a super important potential. It's semi-gut, so it's kind of but it's also semi epithelium, right? And so you kind of get a sample of what might be happening there.
Does anyone else work in these other systems?
I do not work on vaginal microbiome, but it's somewhat related to what I studied during pregnancy and immune regulation. And there have been a lot of studies really underscoring changes of the vaginal microbiome associated with increased susceptibility to vaginal infection and then complicating pregnancy. And also keep in mind that the earliest colonizers in the newborns' intestine actually are derived from the vaginal canal. So any perturbation of the vaginal microbiome would have direct impact on the microbiome in the newborn, and as a result, changing the immune system in the newborn. So, and I would say that's an understudied area. And at the same time, the microbiome in the vaginal cavity is really critical in maintaining the acidity in the vaginal cavity, so that would reduce infection and promote a very healthy microbial community to improve the reproductive health in women. And then in terms of skin microbiome, there's a lot of studies out there and showing. I think that also underscores the discrepancy between mice and humans, because mice will have different skin microbiome than humans. I think that limits the translational power of the mouse studies addressing the skin microbiome and the immune system. Luckily, I think with human microbiome, at least from the skin, it might be easier to sample.
Yep, totally.
Yeah, I'm glad this point was brought up, because on a very simplistic level, it seems to me that increased diversity is a good thing for the gut microbiome, whereas it's a bad thing in the vaginal microbiome, where you go from a lactobacillus dominated community to all these other strains which shouldn't be there. So it's almost the reverse problem where you're trying to control that.
And at least in mouse systems, there's like really strong systemic effects, right? Like the Fishbach paper from a couple of years ago, where they paint the microbe on the mouse skin and it has this, immune response to the engineered antigen. Okay, so I think we're getting close to time, but there's one question here that I think is kind of a good one to end off on. You want to?
Please.
Okay, so this anonymous attendee asks, what do we think the future of microbiome therapeutics will be? Like, how is this going to end up translating into the clinic, into people's lives in like a 50-year from now perspective? What's your sci-fi projection on where we're going to be? Brittany, I haven't heard from you in a bit. Do you want to be the first to jump in on that?
Oh, great. Thanks for that. I mean, we all have the answer we can we can all give the answer of like, oh, postbiotics, prebiotics, probiotics, like engineered strains, or I talked a little bit in my talk where we can change the stuff the microbes have access to, change their enzymes, or change the stuff that they're putting out. Hopefully it gets more elegant than that in 50 years, though, that maybe we've learned a lot from all of these interactions, and we have a totally new way to kind of go about it where it's not independent of the gut, but just more elegant and refined, and we can do it. And I know we often also, you'll hear people say, oh, like personalized medicine, like you get your fecal sample and then that tells you exactly what you need. And it's not that I don't think that that'll be cool or important, but I feel like that's a little bit the way, like Matt, you're saying with the human genome sequencing, like you also heard that, right? Like, oh, get your genome sequence and then you know exactly what disease you're going to have and what drug you're going to need. And I think we're still going to have medicine based on symptoms and broader things than personalized sequencing. But I don't know, maybe we'll come up with some cool stuff. Hopefully it will be pretty personalized, right? Like we don't want mass drugs for everybody that isn't personalized to what your individual state is. But I don't know, sci-fi, maybe we'll just go out of space for a week, go to outer space for a week and get a cure, and then we come back and we've got our space microbiota.
We had our first CRISPR baby, I think, last year, right? So, personalized genomic treatments, maybe we'll be there in 50 years, personalized microbiome treatment.
Giada, I'm just going to start calling.
Did you wave?
Sorry, I was moving a fly, but yeah, but I can jump in. I really hope actually we can move towards personalized medicine and personalized microbial and diet treatment, such as those that, for example, Tim Spector and Aaron Elinav kind of are trying to design for cardiovascular disorders and metabolic disorders. So I'm really hoping, for example, for IBS that we can get there soon. But I don't know.
Yeah, we're at time. I want to be respectful of everyone's timing here. Any final word before we call it?
I think we should let them finish. If anyone else wanted to finish answering that question.
We can go over. But
Yeah, I mean, I'm sure we're going to get there. I'm sure there's too many things that have been lined up. I think we just don't have, if you told me 30 years ago we were going to drug the kinome, I would have told you were crazy. When you told me 20 years ago we were going to drug the epigenetic factors, I would have told you you're crazy. Same thing about the CFTR transporter. We were never going to get that. I love being wrong. Being wrong is the best in science. And so we're going to get there. And when we look back on it, we're going to be like, oh yeah, of course it was going to work, right? Because we have an example to point to. And I think just small molecule therapeutics are going to work because it's really hard to get things to get absorbed from the small intestine. When I talk to drug developers, they're like, you mean you want the drug to stay in the gut? Like I'd have 100 drugs that went through clinical trials successfully if I could do that. If it was an oral drug and it stayed in getting it through the gut is the hardest thing. So I really believe we're going to get there. We just need the one or two examples, the exceptions that prove the rule, and then we'll be going.
Matthew Carter?
I think that many of the reasons why microbial therapeutics have failed to date is because as adults, our microbiome is very resistant to change and is very resistant to outsiders. And I think over time there will be more interventions during infancy when the microbiome is more plastic and more likely to have a kind of sustained change.
Melody?
I think to push the field forward, it's very critical to identify the specific metabolites from microbiome or lipids and the interacting partners from the host sites, and also some geographic localization of the interaction in the human body. You cannot assume that the interaction in the bone marrow, in the gut, would be similar in the lung, in the brain. And also there might be some temporal effect So you cannot just continuously administer this metabolite to a person. You have to identify the intervention window for you to administer. I think all of that would require tremendous research and technology, but I think we're getting there eventually.
Do you have anything to add?
Oh, I'll keep it brief. We've been thinking about digital twins, right? Like if you have integrative models that can take all of that personalized information from a person and build a mechanistic interpretation of where you think the flow of metabolites or what have you are gonna go out, given a dietary context, you can start to make those personalized predictions. But it's such a big, messy, interconnected system that you need some sort of integrative prediction, integrative model to get to that. How about you?
I'm not a soothsayer, but I will just say that I hope that advances in anaerobic microbiology and genetics, as well as just molecular biology, which I do a lot of. I hope that contributes to all of this. And yeah, I mean, we've all sort of prognosticated, and it'll be fun to meet 50 years from now and see how right or wrong we were. But I love Matt's thought about the fact that as scientists, we should embrace um, this idea of being wrong and revel in it in some ways. And I just also like to say that I really missed having June Round at this panel.
Yeah, but
She's a good personal friend.
great conversation, amazing talks.
Yeah.
Thanks so much to all of you. We'll cut it there. I think we're over time.
What a fun symposium. Thank you very much for being there.
Thank you so much.